In the last talk, we discussed some of the problems confronting development of new tools to prevent and to treat tuberculosis. In this next segment, I really want to focus on the problem of antibiotic therapy and delve into this issue of why the antibiotics that we currently use to treat tuberculosis are not more effective than they are. I'll remind you that we discussed in the last talk the Mitchison Hypothesis that's been with us for some time, proposing that TB chemotherapy is problematic because the bacteria inhabit different tissue compartments and therefore adopt different physiologies that make them refractory to one or more anti-TB drugs. This idea, which has been around for quite a while now, might explain why the kinetics of killing with the antibiotics that we use against tuberculosis are in all cases biphasic or multiphasic. So in this case, this is an experiment in which chronically infected mice were treated with one of our frontline drugs, isoniazid, via monotherapy. What you can see, looking on the y-axis at the number of viable bacteria still within the lungs, that's a log scale, versus the days on chemotherapy, is that killing of bacteria by isoniazid, or INH as I'll call it, is not a log linear process. So initially there's a period of rapid cell killing, rapid being a relative term here. It's actually very slow compared to the action of most antibiotics against most bacteria. But the remarkable thing is that after about 4 weeks of chemotherapy, the killing essentially stops and the bacterial population stabilizes at a level about 0.1 to 1% of the initial population. So there's a very high frequency of survivors in the face of chemotherapy. We call these the persisters. We call them persisters to distinguish them from the resistors, which are bacteria that have evolved through mutation resistance to the antibiotics, because in fact this is a phenotypic metastable state that is not inherited by the progeny of these cells. So if you were to take these bacteria out of the tissues, regrow them in culture, reinfect animals, and treat them again, you would get exactly the same curve back. They are not stably, heritably resistant mutants. This is a purely phenotypic phenomenon that we know very little about. But clearly, if we could convert this biphasic curve to a simple log linear kill curve, that would be a major advance towards accelerating the clearance of infection with chemotherapy. Now, we can postulate that the reason for these biphasic kinetics is due to the existence, as Mitchison proposed, of a dormant subpopulation of bacteria that is killed with slow kinetics compared to the bulk bacterial population. If we perform a simple mathematical model in silico for this process on the basis of differential killing dynamics of a fast population, that is a population that's killed with rapid kinetics, and that represents the bulk of the bacteria, maybe 99.9% of the bacterial population, versus a slow population, the persisters, that's killed with very slow kinetics, and then calculate the composite of these two curves as shown by the blue line here, what we get is an almost perfect match to the experimental data. Now, of course, the fact that a model matches the experimental data does not prove that it is true, but it at least says that the idea is plausible. So according to the Mitchison hypothesis then, the persistence of TB in the face of chemotherapy is due to a set of environmental inputs leading to a phenotypic switch, in this case a state of dormancy in a subset of organisms, which causes them to become these drug-tolerant persisters. So there's a simple flow of information from the environment to the physiology of the bacteria, and this is what's responsible for the recalcitrance of these bacteria to killing with antibiotics. It's a very attractive idea, and as I indicated in the last talk, there's actually some clinical evidence in favor of this idea, but there are some awkward bits about this model as well. One of the most obvious and glaring being that the persister phenomenon is not peculiar to the in vivo environment. In fact, even if we grow a clonal population of bacteria in an axenic culture, just in a tissue culture flask in vitro, and then subject them to a drug like isoniazid, we see that the killing curve is at least biphasic, if not multiphasic, just as it is in vivo. Now, the kinetics of killing are quite different in vitro as compared to in vivo. I don't want to say they're the same, but the biphasic shape of the kill curve in vitro is essentially the same as what we see in vivo, suggesting that the persister phenomenon, although it may be modulated by the tissue environment, is not absolutely dependent, perhaps, on the tissue environment. In a way, this is good news because it means that we can study this phenomenon in a test tube rather than in an animal, and that, of course, greatly accelerates the research on understanding this persister phenomenon. So the fact that there are no obvious environmental inputs in this azenic culture for generating a heterogeneity of the population suggests that instead purely stochastic factors, random fluctuation from cell to cell in gene expression, interdivision time, and so on, could play an important role in generating the persister phenomenon. So we would like to postulate that stochastic processes may in fact underlie the fundamental phenotypic switch to the drug-tolerant persister state. Now, this does not, of course, preclude a role for environmental factors. In fact, environmental inputs could have a modulatory effect or a stimulatory effect on the rate of switching, even if the underlying switch mechanism is essentially stochastic. And there are excellent examples of this from model organisms that have been well studied, probably the best studied of which is the switch that occurs at the lac operon in Escherichia coli. So I'm sure all of you have studied the lac operon at one time or another in your undergraduate studies. I'll just remind you of the components of the system. The lac operon encodes a beta-lactamase, coded by the lac-Z gene, a permease, lac-Y, that brings lactose into the cell, and the beta-galactosidase can then act on that lactose that's brought into the cell to convert it in part to allolactose, which is the true inducer of the system. Allolactose then binds to the lac-I repressor and causes it to fall off the DNA, thereby de-repressing expression of the lac operon. So again, the sequence of events is lactose enters through the lac-Y permease. That lactose is then converted to allolactose by beta-galactosidase. That then causes de-repression by binding to the lac-I repressor and causing it to fall off the DNA, leading to de-repression of the operon. So it's a very simple, lovely architecture, but it obviously can't work, as I've drawn it here, because in an uninduced cell where the lac operon is repressed, there's no lac-Y. There's no permease to bring the lactose into the cell. And even if the lactose could enter through, say, simple diffusion, there's no beta-galactosidase to convert that lactose to the true inducer, which is allolactose. The reason why this system works is that, in fact, it's not perfect. It's leaky. In a subset of cells, there's always some expression of the lac operon, and this is simply because repression depends on the continued interaction of the lac-I protein and the lac-O operator on the DNA. And like any intermolecular interaction, it breathes a bit. Sometimes the lac-I just falls off randomly, and when this happens, there's some transcription of the lac operon leading to some production of lac-Z and lac-Y, and that subset of cells in which these events have occurred are now primed to respond if lactose happens to be added to the culture at this time. So there's always a subset of cells, even in a clonally homogeneous population, a genetically homogeneous population, that are primed, if you like, purely by accident to respond to lactose if it should happen to enter the environment. The key, as I said, is lac-I and the interaction of lac-I with its operator. Now, it's important to note that although this is essentially a stochastic switch that occurs when lac-I happens to fall off the operator, the rate at which actual induction can occur in switching to the lac-on state is modulated by environmental factors, most notably by the presence of other sugars like glucose in the environment. Glucose is a preferred sugar over lactose, so when that is present in the environment, even with addition of lactose, the rate of switching is modulated because glucose blocks the uptake of lactose, and it also represses the CRP transcription factor, which is required for expression of the lac operon. So here we have an example of what is fundamentally a stochastic switch to a state that's primed for responsiveness to lactose, but where that switch, that phenotypic switch, can be modulated by the presence of other environmental factors like glucose. What this kind of architecture then leads to is a bistability of the response of the bacterial population to an inducer like lactose. When lactose is added, those cells that are primed to respond completely and rapidly, and they induce the lac operon to its full state. In those cells in which lac-Z and lac-Y are still absent, when lactose is added, they're essentially blind to it because they can't take up the lactose and they can't convert it to allolactose, and so they stay in the uninduced state until by random chance, lac falls off of its operator and allows those cells to become primed. And because this is essentially a positive feedback loop, once cells have entered a state in which they're capable of being induced, they are very rapidly induced to the full state. So in a situation like this, as shown graphically here, if you look at a population of cells after addition of an inducer like lactose, where the cells have been engineered to express GFP once the lac operon is turned on, that's the green fluorescent protein, a marker that can be used on a single cell basis, what you find is that the population of induced cells consists of two discrete subpopulations. These white cells are cells that were not primed to respond to lactose at the time this inducer was added, and therefore they have not. So the lac operon is still off in these cells. In contrast, these green cells were primed to respond, and they have by completely inducing the lac operon. There's no intermediate state. So in fact, cells are on or they are off. They are not partially on or off. So this is a bistable phenomenon, rather reminiscent perhaps of the persistor switch leading to a subpopulation of cells that are refractory to chemotherapy, although in that case we do not understand the underlying mechanisms. And this bistability of the population that I've shown you, where simultaneously in a clonal population two very different cell types can coexist, I think underscores the inadequacy of the traditional approaches that many of us have been taking to study phenomena in microbiology and in cell biology and so on, based on the averaging the behavior of cells using what I call batch culture methods. So that would typically be to take a population of cells growing in a shaker flask like this one, and then to pool the cells, to grind them up, to extract, say, an enzyme and measure enzyme activity or the amount of DNA or this sort of parameter. These approaches are very good for giving you an average value, which I'll call alpha, of some parameter in the population. But these approaches reveal nothing whatsoever about the underlying population structure, which can be quite heterogeneous and which, for the same value of alpha, can represent very different underlying population structures. And that's illustrated in simplified form graphically here, where I have schematized on the y-axis number of cells occupying a particular phenotypic state versus on the x-axis some particular cell parameter. That could be expression of a particular enzyme, particular cell growth time, interdivision time, any parameter that can be quantified. So my point here is that in these four different populations that I've illustrated, the average value alpha is exactly the same for these four populations, and yet clearly these are very different populations of cells. So this average value alpha could represent a simple narrow Gaussian distribution, as illustrated in this panel, or a broad Gaussian distribution, as indicated here. It could even at one extreme represent a bimodal distribution of cells, in which the average value, in fact, is not represented by any individual cell within the population whatsoever. So that average value is extremely misleading vis-a-vis the actual phenotype of individual cells within the population. This bimodal or multimodal distribution can also occur with the interesting phenotype, if you like, being represented by a small minority of the population. This would be representative, for example, of the persister phenomenon, in which the cells in this small bump here are the ones that we're actually interested in. And yet, if we were to take a batch culture approach to studying this phenomenon, this is the signal we would get, the signal from the cells that we're not interested in, and we would learn nothing about the behavior of the subpopulation of cells, which is in fact the target that we want to study. So we and many others now are taking new approaches to the study of bacterial behavior at the single cell level. That is, approaches that will allow us to capture the full complexity of cellular behavior and the variation between cells in behavior of particular characteristics. One of the approaches in my lab that we're taking is to use a combination of microfluidic culture of bacteria and time-lapse microscopy to study the behavior of large numbers of individual cells before, during, and after imposition of a stress, such as antibiotics. We do this, as I said, using microfluidic devices that we fashion using soft lithography, and which are illustrated schematically here. These consist, in this case, for example, of a simple glass cover slip on which the bacteria are seated, which is then overlaid with a semi-permeable membrane, so that the bacteria are growing right underneath the membrane, sandwiched between the membrane and the cover slip. We then overlay this with a block of material that's been fashioned by soft lithography, with an input port for media to flow in, an output port for media to flow out, and flow channels cut through it for the media to flow across the surface of the membrane. So this is essentially a sandwich through which we can pump media that feeds the bacteria. They grow happily on the cover slip, fed by the material diffusing through this membrane, and using a fully motorized and computer-controlled microscope that can visit as many as 100 different points on the cover slip and remember where it's been so it can keep coming back to those same points time and time again. We can collect over weeks of observation, even months of observation, in fact, the detailed behavior of a bacterial population at single cell resolution. So first and most importantly, we find that when we grow bacteria in these microfluidic devices and then expose them to a drug like isoniazid, the kill curve that we see is clearly biphasic. There's a period of rapid killing followed by a period in which killing essentially stops. So the phenomenon that we're trying to study, this biphasic killing response to the drug isoniazid, can readily be studied at single cell resolution using these microfluidic devices. So the beauty of this approach is that it allows us to study the genealogy, if you like, of individual cells. We can follow them by recording their behavior through multiple cell divisions before we impose a stress such as the drug isoniazid. We then expose them to the drug for whatever period of time we desire, which of course leads to the death of most of the cells, with the exception of the rare persisters. But then we can readily identify these persisters as cells that fail to lyse and which recover growth after we wash out the drug. And now we have a complete record through time of the behavior of these cells and their ancestors. So we can now ask, is there anything that we can identify that's different about these cells, either before, during, or after the exposure of the drug, that causes them to be persisters when 99.9% of their siblings are not? So using this type of approach, it was possible for the first time to test an hypothesis that was proposed many years ago, in fact, more than 60 years ago now, by Joseph Bigger, in a classic paper where he looked at the response of Staphylococcus, in fact, to treatment with penicillin, again, in a test tube, and for the first time observed this persister phenomenon and coined the term persister. He suggested, it was purely hypothetical, but it was quite a brilliant insight, in fact, that the persisters might be insensitive to the penicillin because they might be temporarily in a dormant or non-dividing phase, similar to bacteria that have exited the cell cycle due to, say, nutrient starvation. And this would make sense because penicillin kills bacteria only when they're undergoing active cell growth and division. So that was Bigger's hypothesis back in the 1940s. It had to wait 60 years, in fact, for experimental validation using newly developed tools for time-lapse microscopy and microfluidics. And this was reported in a paper published in the year 2004 by Balaban and colleagues. There's the reference for those who are interested, in which they used microfluidic cultivation of Escherichia coli to study at single cell resolution the response of these bacteria to the drug ampicillin, a beta-lactam. So the persister in this case is circled here in red. So I'll ask you when the movie plays to focus on that cell. As you can see, the cells around it are actively growing and dividing, but that pair of cells is not growing. Ampicillin is added and the cells die, but when it's washed out, these cells are capable of resuming growth. And in fact, if you follow them long enough, they eventually resume rapid growth that is indistinguishable from the growth rate of their siblings who died in response to the drug. So using this type of approach, Balaban and colleagues were able to demonstrate that Bigger's hypothesis in fact was correct, that the persisters comprise a pre-existing, and that's the important point, this is not an adaptation to the drug, it's a pre-existing subpopulation of bacteria that are temporarily in a slow or non-dividing state and are therefore protected from the killing action of this drug. The actual mechanisms that are responsible for generating these persisters are likely to be stochastic, but they have not been identified. Likewise for reactivation of these persisters after washout of the drug. So that's a major challenge for the future, is to try to understand the underlying molecular processes that allow these cells to become persisters. But at least in the cellular level, in the case of E. coli, it seems to be very clear that Bigger in fact was correct. Persisters are a pre-existing subpopulation of non-growing cells. So when we began to collaborate with this group to study using these same approaches, the persister phenomenon in Mycobacteria, focusing at first on the drug isoniazid, we had a rather strong bias at the outset that the persister phenomenon in mycobacteria was going to be very similar for the reasons I've already described, and which Mitchison proposed, namely that drugs like isoniazid kill bacteria only when they are actively growing and dividing, and therefore it would make sense that the persisters would be these cells that are in a temporarily non-growing state. So that was our assumption when we began these experiments, or our hypothesis, rather, when we began these experiments. But the beauty of doing science is that our ideas about the world are never as interesting and complex as the world itself. And so we found, in fact, very quickly that this idea was wrong and that what had been discovered in E. coli, in fact, apparently does not apply to Mycobacteria. That's shown here, a typical experiment in which we have grown Mycobacteria, in this case, in a microfluidic device. The bacteria have been engineered to express GFP, as in the previous E. coli experiments. That's why they glow green. So what you're going to be looking at is bacteria grown in 7H9 medium initially, which is just basic broth with no drug added. We'll then switch to isoniazid containing medium. That'll be shown by the INH appearing in the corner there. And look at the response of cells to the drug. We will then wash out the drug. 7H9 will reappear, and we'll look at the regrowth of the rare persisters that survived exposure to the drug. So if we can roll the film, you see cells are actively growing and dividing in absence of drug. As soon as we add the drug, they cease growth within minutes of addition of the drug. It's really an extremely rapid response, and then the cells undergo lysis. So they stop growing very rapidly. Then there's a delay of about a day or so before cells begin undergoing lysis. This particular experiment involved recording the bacteria for a period of about 1 week. So this is a very compressed time course here. Now, remarkably, after prolonged exposure to the drug, this subset of surviving cells starts to grow and divide again at slower rates than in the absence of drug, but at very appreciable rates. Some of the progeny of those divisions die, some survive. Those that survive long enough can then, when the drug is washed out, reactivate growth, and in fact, they do so immediately, and they resume very rapid cell growth within just a few minutes of washout of the drug. So the response of the bacteria in this case is very different than what was seen by Balaban and colleagues in the case of E. coli responding to the beta-lactam ampicillin. Now, using this type of approach, we can, as I said, study the response of bacteria at single cell resolution, and this has made it very clear to us that in fact the behavior of these cells is even more heterogeneous than we might have thought. That's shown, one example of this in the movie shown here, where you're going to see a rare, rather atypical cell, but one that we think is behaving in an extremely interesting way that we think is likely to be very informative about how the drug is actually acting on these cells. So we could roll the film. What we see again is that cells are growing and dividing in 7H9. We add the drug, they arrest very rapidly, they swell a bit, and then they begin to undergo lysis and disappear from the frame. But focus on the cell over here. It's going to do something rather extraordinary. It arrests, but then in the continued presence of the drug, it eventually resumes very rapid cell growth. In fact, it's growing as fast now as though there's no drug present at all. But clearly, this was not a drug-resistant cell because it did eventually die. In fact, it appears that division is inhibited in the cell, even though cell growth is not. So my point in showing this sort of rare occurrence is that there's an enormous amount of heterogeneity in the behavior of individual cells within a bacterial population that simply isn't captured by the kinds of batch culture methods that we typically use to study bacterial behavior. My personal feeling is that the future of microbiology is going to be to focus on the individuality of bacterial behavior rather than to stick with the averaging methods we have used in the past. And I think this is going to be a particularly important approach for us to take when we want to study the behavior of important subpopulations like the persisters. So there are a number of points here that I want to make in terms of compare and contrast between E. coli. The first and most obvious one is that in contrast to E. coli, where cells that turn out to be persisters after drug exposure are already in a very slow or non-dividing state prior to the addition of drug, this simply isn't true in Mycobacteria. So in fact, if we quantify the growth rate prior to drug addition of cells that die over here versus cells that survive over here in a particular experiment, these are data from one experiment, we find that there's absolutely no difference in the average growth rate or in the spread of growth rates between these two populations. So Bigger's hypothesis apparently is true for E. coli responding to ampicillin, but it is not true for mycobacteria responding to isoniazid. We do not at this point know whether this is a difference in the species that we're looking at or a difference in the drugs that we're looking at. This remains a challenge for the future. So that's the first take-home lesson from this sort of compare and contrast, that in E. coli, persisters are rare, slow-growing cells, and they exist prior to antibiotic exposure, whereas in Mycobacterium, the persisters are growing and dividing at completely normal rates prior to antibiotic exposure, so it's a very different phenomenon. The second point of contrast is that in E. coli, the non-persisters, those cells that actually die in the presence of the drug, continue to grow at nearly normal rates after exposure to the antibiotic. So the antibiotic doesn't stop them at all. They keep on elongating until they lyse. And that cell lysis occurs actually quite rapidly after exposure to the drug. In contrast, in Mycobacteria, the non-persisters, that is the bulk of the population, arrest growth really within minutes after exposure to this drug, isoniazid. So it's a very rapid response to the drug. But then cell lysis begins only after quite a prolonged delay of about 24 hours or so after initial exposure to the drug. So it's a completely different, really opposite response to drug exposure. In E. coli, after washout of the drug in the microfluidic device, the persisters do not, in the continued presence of the drug, the persisters do not resume growth as long as we continue to pump it through the microfluidic device. In fact, they do not resume growth until after washout of the drug, whereas in Mycobacterium, the persisters, as you saw, resume growth and division after prolonged, that is, many days of antibiotic exposure, although some of the progeny of those divisions, in fact, do not survive. And when we wash out the drug in the microfluidic device, we find that E. coli persisters reactivate with slow and stochastic kinetics after antibiotic washout. So it's almost as though it's a process of radioactive decay determining whether a cell can reactivate at a given time or not after drug washout. Whereas in Mycobacteria, those persisters reactivate almost immediately, very rapidly after drug washout and with uniform kinetics. So it seems very clear that the drug is actually keeping them in this non-dividing state. As soon as that pressure is removed, the bacteria start to grow again.